Welcome to the DNA Talks podcast, where we take on the mission of unlocking the code of your genetics. This season is all about you, upgrading your health not just on the surface, but down to the root cause. Join us as your clinicians at the DNA company investigate your DNA and beyond. The intention of this podcast is to enhance your lifestyle by changing what is in your control. This does not substitute the medical advice given by your personal doctor, therapist, and other healthcare professionals. Good evening, everybody.
Good
I guess
evening.
everyone's settled in now. And here we have this fine gentleman. Look at this amazing man that we have here. The blessing we have with us tonight.
Oh,
Dr.
thank you.
Ghannoum. Thank you for coming on and taking your evening to share with us.
It's my pleasure, my great pleasure to be with you.
No, it's amazing because people might not realize the work you've been doing, but they have heard us talk about this subject, and there's no better person to guide us down the path than you. And the subject has always been, hey, you guys do DNA testing. Do you look at the gut? And our answer has always been that we believe the gut is the second half of personalization. DNA is your innate wiring. It's your cellular instruction. But there's a whole other ecosystem of DNA living in your gut. And if you start to understand what's actually happening there, and it's separate from you, there's more DNA in your gut than there is in your body because of all of what's going on the flora, all of the bacteria, right? So if you start to understand this, what this looks like, and you pair that with your DNA, the view that you have of your body, there's really not many stones left unturned, right? Yeah. So when we've been working with people, and everybody here is probably familiar, there's been this question of, wow, I learned how my body works. I learned how my detox system works. I learned how my brain works. I learned how my hormones work, which is so implicit in so many areas where people get stuck. And this question of what do we do about the gut? Because I feel like there's just still something left. And so our answer, like I said, has always been we believe it's the other half of personalization, but we never had a tool that we thought did the job. And that was always where we were stuck. It's very easy for us to just go test people, right? Just like anybody would have experienced in what we do with genetics, the reporting and the interpretation is much more functional, meaning it's not, here's your DNA, good luck. It's, we think you need to work on your hormones, and very specifically in this little nodule of the cascade, because this is where there's a failure. And guess what? Here's exactly what to add. Here's exactly what to remove. Actionable information. And this has been missing from the gut for some time. You've probably seen apps and products that tell you eat less broccoli, exercise 3 days a week. And sometimes you happen to hit the nail on the head and it just works. But the majority of the time you hear people saying, this sucks. Like truly, that's the feedback that we get. This sucks, this doesn't work, the gut microbiome doesn't matter. It does matter. The tools have not been functional. The tools have not looked in the right areas. It just like people ask us the question, what's the difference between the DNA company and 23andMe? The DNA company is looking for functional pathways. We're looking for less than 100 genes that actually matter and then telling you what to do about it. 23andMe is saying the majority of what we can sequence, or the more that we can sequence out of the 22,000, the better we're doing. Data, 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 as opposed to interpretation and insights. Data is dumb unless you know what to ask it and get some feedback. This is where we've been stuck. So thank you again, Dr. Ghannoum, for joining us. And it's amazing that you're going to walk us through this. He's actually brought a report of an actual patient to show you how it's possible to interpret in the gut microbiome actionable items. So firstly, let's just have a backgrounder. Why do we need to know? Why does it matter?
So really, first of all, thank you very much for really very nice introduction. And I love the fact that you are talking about the DNA and the microbiome, how they make the total story instead of one part of it. Now, why does it matter that we should look at the microbiome and our gut? Because, recent advances in science, especially this next generation sequencing, allowed us to look at organisms or microbes that live in our body, on and in our body. So, like we have microbes, as you know, in our hands, on the skin, in the hair, all over the place, but particularly in the gut. And really, what you mentioned about the number of genes there compared to what we think we have. Like, for example, we have 23,000 genes in the human. When you look at the number of cells, we have 600,000 genes. So really, in a way, you have another world there, as you alluded to, that live inside us and that can play a role in our health and disease, in fact. And this is what we call the microbiome. For years, people defined the microbiome as the bacteria that live in our body. But now, with the advancement of science, we know that it's not only bacteria, there is fungus, as well as there are viruses also present in the same environment. And we published a number of studies where we showed that these guys are not just sitting together separately, they are in a little like sandbox. They interact together. So when they are good, it's like when children plays in a sandbox, everybody, the parents are happy. But then you come and put a bully there, and in this case, the parents, and especially for me as a granddad, you will be upset because the whole environment is disturbed. And that's what happens in our gut. So really the big picture, why we should know, because when this, as I mentioned, the balance of the microbes that live in our body are mainly beneficial microbes, such as lactobacillus, bifidobacterium, then our health is good. However, if we have an increase in the pathogen, in other words, organisms that can cause disease and are resistant to a treatment, then we will be in what we call a state of dysbiosis, which is imbalance of the microbiome. And when you have imbalance in the microbiome, it not only will affect your gut, like, for example, different gastrointestinal symptoms, but also could affect our other parts of our body through the what we call gut-brain axis. These microbes in your gut, they communicate with your brain, and they could affect anxiety, depression, all these neurodegenerative diseases. So that's why it's very important for us to look at the microbiome.
And I mean, you mentioned, lactobacillus and bifidobacteria. These stand out as things that people talk about. And it also, the fact they pointed those out, people look at those and it really gives you an idea of what's important about the gut. Because if you look at a 10-year-old and a 15-year-old and a 30-year-old, you're going to see this decline. These two very healthy strains that you need, that, why does every 15-year-old, well, not every, but the majority of them, look in better shape than every 30-year-old, right?
Yes.
There's this fat burning situation going on in the gut if you have the healthy right blend, but the beneficial stuff seems to deplete over time. That's why it's not just knowing, it's also knowing what good stuff you're missing and what bad stuff you have a little too much of. And then that drives the action. So why does this equal disease? When you said, like, I get it, there's an imbalance. The levels should be like this, or they're more like this. Why does that equal disease?
Well, I can tell you why. First of all, in addition to the lactobacillus and the bifidobacterium, I want to add the yeast, Saccharomyces cerevisiae. It's also a good guy we need to have, unlike having Candida, which is the bad guy. Okay. So why it causes diseases? Because these organisms, especially when we have less of the good guys, the beneficial microbes, we have these pathogens, they can come together. And as I mentioned, they interact. And what they do, they start affecting our gut lining, okay? And then they cause what you call perforations or a breakdown of our gut lining, causing leaky gut syndrome, where things can go out, that toxins, other bacteria, other microbes can go into our bloodstream and start causing issues. Also, what they do, a lot of the time, they encourage pro-inflammatory cytokines. In other words, they increase the inflammation. Compared to when you have the good guys, you have more anti-inflammatory cytokines. So that's why it's very important to look at them, because what we would like to do is to increase the beneficial one. Why? Because like the pathogens, they secrete some molecules, we call it metabolites, small chemicals basically, such as short chain fatty acids, which have been shown to help us in our immunity, also help us in gut-brain axis communication. So that's how really the totality of things work.
And, so you mentioned in there, like, we need to increase. You make it sound easy. Does that mean that once you identify something, you know what to tell someone in order to get that increase?
You know, this is very, very good question because and it's complex, as you said. It's not very just take, let's say, a probiotic and then you are going to be fine. No, what you need to do, you have to follow a holistic approach. Once we know, look at your gut, okay, we see that there is imbalance. We also look at other factors, your lifestyle. Are you stressed out? And as you know, these days we are all stressed out. Also, what type of diet do we eat? Like, you know, the westernized diet have been shown to really have a bad effect on our microbes living in our gut. So all of this you need to start looking at your lifestyle. Do you sleep well? What sort of things you eat? And this is all you need to put it together. Like once you do the testing, you find there is imbalance. We look at the factors that are making or leading to this imbalance, and we can recommend what sort of things you need to do to bring it back into balance and hopefully feel much better.
So for everyone that's listening, this probably sounds familiar, right? This probably sounds like, wait a second, that's what they said about DNA. The truth is, it is the same thing. What we're saying is the gut and your genes will tell you where to prioritize, right? We're not diagnosing a disease. We're not saying that you have a propensity. We're saying here's a certain job, certain function that is just not happening because there's an imbalance, whether in your gut and what's supposed to be there, the community, the ecosystem, or there's a certain gene that's not active. Because when we look at, and again, everybody here will be familiar, when we look at certain detox genes, we find that people that are missing some of the key detox genes of the gut end up in the exact same place. Gut wall permeability, dysbiosis, leaky gut, leaky brain, mood issues, etc. It's the same outcome, different input that caused it. So now imagine you fix that and you get to 80%, right? And you're probably going to, in that path, also fix your gut microbiome just by nature of fixing the gut wall itself, you know, that it creates a balance. But there's a little tweaking left to do. And that's kind of what we're saying here is that if we get this right also, there isn't much left to do, right? And then you've got yourself to your optimal version. So with that, I know you brought a report.
Yes.
So we're going to walk through it. And I've been excited about this because I'm learning from you, and you know, and there's things that you know that just aren't out there, functional insights. So let's walk through the report as if maybe I'm the patient. Talk to me, right? As if it's me. And I promise I'll do what you say.
So then we need to really look at the report. You have a version of it, which maybe we can share also down the line. But really what this report does, it looks at you, in a way, personalize your gut microbiome. What microbes, both bacteria and fungi, and this is very important. It's not only you look at bacteria, we look at also at the fungus, as I mentioned, because these are play together, and the studies have shown that there is really differences. So what we look, you send a sample, you also fill a questionnaire so that we can capture your lifestyle. If you exercise, how often you do, if you sleep well or not, if you are stressed, what sort of diet do you eat, you know, all of this, we put it all together. But what happens at the beginning, the sample comes, you fill the questionnaire before that, and then we start looking, we do the analysis in the lab, we do the sequencing, and we come out with a report that describe a number of things. Number one, it tells you what is the diversity of the microbes that live in your body. In other words, you know, like in our society, I am always think a society that diverse is much better than a society that is only one type of people live in. We are all human beings. Once we are diverse, it really works very well. The same could be said with respect to the microbes that live in our body. The more diverse they are, the better your gut health as well as overall health will be. And that's one factor we look at. What different types of organisms? The more you have, the better. Okay. Then we look at what sort of beneficial organisms are there. And two things we do. We identify the beneficial organisms. Are they there or not? But importantly, we tell you what is the abundance or the level of these organisms. We have done studies and we found that really there are we looked at about 5,000 healthy people and we looked at their microbiome. what I mean by healthy, we followed the Human Microbiome Project, people who don't have a disease, don't take drugs, basically a general health. We eliminated all or excluded these people who have issues, and then we start looking at healthy people. And this allowed us to, for example, if you talk about E. coli, a lot of people hear about the bacteria E. coli. We look at in healthy people, there is a certain range between this level to that level, you are healthy. If you are above that, because this is a pathogen, then we know there is an issue, we need to bring it down. So we look at the beneficial organisms first, and we see, do we have enough of them, or we really don't have in certain, like for example, you look at bifidobacterium, not a lot of bifidobacterium, and we look at specific ones, and then we say, okay, we need to increase this, okay? Now, we look at also lactobacillus, we look at other organisms which are good, okay? So we look at, such as I mentioned, Saccharomyces cerevisiae, which is the yeast. It's good to have it. We and others found in healthy people, they have high level. And of course, I told you there is the range. We know the range now because we looked at these 5,000 people. And if you are healthy, you should have a range between this and this. So this way we define, do you have the right beneficial organisms? And of course, if you don't have, that's where in the recommendation we start to say, okay, how can we increase that? Okay. Also, so we looked at diversity, we looked at beneficial organisms, but also we looked at pathogenic organisms. In other words, organisms that can cause disease. I mentioned E. coli, Clostridium, C. diff, for example. And in the fungal world, do we have the Candida, which is, as you know, a lot of people know about Candida, that it could cause issues, okay? But it's very important to know the level, because Candida at low level, low abundance, is quite good, okay? Because 70% of us in this call, they have Candida, as long as it is not overgrown, okay? So that our testing will tell you whether it is out of balance. In other words, you have more than what we really would like to have, okay? Then we looked at, do we have, for example, anti-inflammatory organisms that have that, like Faecalibacterium proteinsiae, for example, is an anti-inflammatory organism. So we need more of it. How can we encourage it? However, if you have Protobacteria, the phylum Protobacteria, this is a red flag for inflammation. So again, we look at that and say, oh, this is elevated. In other words, the abundance is high compared to the healthy people. We need to fix it down. And there are ways to do it. For example, using vitamin D3 and fiber. So we then can customize the recommendation based on all this report. At the same time, we look at your 50 questionnaire with respect to exercise, stress, and whatever. I can tell you a story. One lady, she sent her fecal sample. We analyzed it. It was really out of balance. So we looked at what she is eating, and she really was eating very good food. It's not like hamburger every, breakfast, lunch, and dinner. But what we found, that she was highly stressed. So then we start to recommend to her, look, you need to do meditation. Is very important. We are all, at work. If you are a lady, you are at work, you're taking care of your kids, you come home, you need to cook also. It's really stressful life. So we need to start to recommend doing meditation. Try to take 15 minutes a day and, relax a little bit. Don't think about anything. Also yoga. And I tell you here, it's so funny. I come from Lebanon originally, so I never did yoga in Lebanon, okay? Now I'm starting to do yoga. Why? Because it relaxes you. You go, I go on a Sunday, like 1.5 hours. All what I'm trying to do is, can I flex myself? I don't think about anything else. And by the way, women are smarter than men in this regard, because when I went to the class, I was the only male there. So now when I give a talk to, in symposia, I said to my male colleagues and the gentlemen, we need to learn this. You should have some yoga. Okay. So we helped that lady, and that really helped her. So you can see is a total really approach.
I remember reading from Dr. Tammy Maragula, who I think is on the West Coast. You have this problem of stress. So when it comes to heavy metals, chemicals, hormone issues, we know how to detoxify them. We know that they're toxins, and we know how to detoxify. When it comes to stress, we don't really know. And really, the detox for stress is meditation. That's how you actually cleanse your cells of the damage done. Deep breath work, and that's the best way. So, even that one little tidbit, this is where, again, people that get stuck, I'm doing everything right. What happened? Like, I'm eating right, I'm exercising right. And seemingly they probably, everybody else, their clinician might even think they're right. Could be that one little thing. That's what I'm saying, that the precision around finding the red flag, whether it's in your genome, whether it's in your gut, that then tells you the story, why everything's out of balance, right?
Absolutely.
Yeah, we've been excited about this for a long time, because you can hear in what Dr. Ghannoum's saying that he's interpreting the data. He's not just talking about this bacteria means this bacteria means that. He's also telling what the outcome is and then what to do about it. And this is what's been missing from gut microbiome testing for some time. And this is why we're so excited to collaborate. We put these two things together, which we are doing now. You can imagine the power of that, right? So I'm going to share the report. And this is Jonathan's gut report, right?
Yeah.
So when I see this, so there's it's exactly what you said: microbiome diversity, beneficial species, pathogenic, and what is the phyla balance?
Phyla balance is you look at the bacterial phylum and the fungal phylum, which means people know about E. coli, which is a species. Okay, so you go to higher level, which is the phylum, which gives you, we did a study where we looked at different what you call enterotypes, and we found, and that's at the phylum level. Okay, and it tells you that your phylum is really, as I mentioned, too much protobacteria. This is, or too much what you call in the fungal world, you look at Ascomycota is a phylum. The Ascomycota, which has Candida, which has Saccharomyces, and this sort of thing. So you look at, this is a higher taxonomic level before we go down into the species and the genus.
So this particular person got a 2 out of 10.
Yeah.
And they got a 1 out of 10 on their phyla.
Yeah.
And so when you see this, what's the first thing that comes to mind in terms of work they need to do?
Then we the first thing we look at, okay, what are the phylum that are down? How can we increase? Like, for example, you can see here protobacteria, it's really bad. So we need to bring this down. So then we can start to advise what sort of steps you need to do to bring this down. Also here, we need to increase it. Like, for example, Basidiobacterium phylum, we need to increase it. So we need to move it into the green zone. If you are in the green zone, then you are in a good shape. Okay, so we start saying, okay, this is what you need to do to bring the Bacteroidetes into the green and the Firmicutes into the green. Like, for example, Firmicutes, a lot of people who are obese tend to have high level of Firmicutes, okay? And people who are, as I say, with inflammation, they have a lot of protobacteria. So what to do? I give you another example about the protobacterium. We did a study with sweeteners, artificial sweeteners, and we published it, where we showed that mice, it's in vivo study, mice that consume a lot of sweeteners, they, guess what? They have an elevation of protobacteria. So to me, look, if you don't, we need to cut sugar, because that's another story which we can talk about. But we need to make sure you don't take sweeteners, because this is going to increase the pro-inflammatory microorganisms, and that's not good. So then we tell you, avoid the sweeteners, take fiber, take vitamin D3. So this is what we look at.
So when you see this and you see the proteobacteria is high, the other two are low, and then you come up with an action plan, can we do the repair all at the same time? Or is it like we need to work first on proteobacteria and then the other stuff?
So what we do in our recommendation, we'll try to say, look, you have elevated this person particularly is really a very poor gut health. Okay, so we try to, as I mentioned, for example, let's say with ascomycota, it's high, but not as bad as but we can fix it. So what we'll say to try to reduce, for example, candida, don't eat sugar, take some proteins and this sort of thing. To reduce protobacteria, we talk about the fibers and vitamin D3. With respect to firmicutes, we talk about, are you overweight? Sometimes, women, when they are pregnant, they have high level of firmicutes, but they need it because they need the nutrition, the absorption, but if you are obese, then we start to guide you towards exercise. So we try to address each one and come out with a total plan how to do it.
Got it. So now we're looking at beneficial species.
Yeah.
As you said, the good guys, right?
Yeah. Yes.
This is stuff you want. And now when things are out of range, like this person has 0, here, and they got a 1.35, like they're really not doing well. So
yeah,
how do you take something like this? And like you said, it's not just a probiotic. So where do you get them from this really bad baseline to somewhere better?
Yeah. Like, for example, Faecalibacterium, okay? This is an anti-inflammatory organism. You can see it here. It's low. It's 1.35%. We need it higher. Now, this is very interesting organism, okay? And that's where the NIH funding that we have really helps us to try to find ways to try to fix these things. And that's what we use in the recommendation. Faecalibacterium, we all have it, but if it is low, you have some issue because you lose its anti-inflammatory properties. One of the things we tested, we are about to publish a paper to look at what ingredients, like prebiotic ingredients or other ingredients, that will allow the Faecalibacterium to increase, to grow better in our gut. Because we have it, we just need to give it more nutrition. And now we have identified some prebiotics, or some of them fibers and this sort of thing that will increase this. So you can see we can really customize based on what you have.
Doctor, I have to tell you that what you're doing, a lot of companies, and we know of some, they come out with a product and it's a business, so they come out with a product that's profitable, and that's kind of where things end. And it's funny because when we first started speaking to you and your team, you were always busy in research. I'm like, why? What is this guy researching? Because the product's been live for a while,
Yeah.
And it's because there's never an end to learning.
Sure.
Right. And there's so much more. And this is what I love about what you're doing, that down the line, we're going to keep learning and learning because you're going to learn and learn, and the product's going to keep getting better and better. Right. And this is awesome because we don't see this when it comes to science and product. They get separated. Somebody will go pick up some science from somebody and commercialize it, and it ends there.
Yeah.
But you don't stop, but it's awesome.
What's so interesting? People come and tell me, are you going to retire? Because I'm tenured professor at Case Western. I say, no, I don't want to retire. I really like what I'm doing. I enjoy it. Sometimes, like all of us, sometimes it's frustrating. You have to keep after, do this. But really, I have funding from the National Institute of Health to look at interaction between bacteria and fungi in the gut. Okay, so this funding, like I got, this grant was $3.1 million to investigate this. So that's why all the time we are doing more studies to try to say, okay, how can we fix this? How can we learn more about this? And that's what it really keeps me happy and excited, because a lot of the time as an academician, not always your research can be translated into good helping people. But when it does, it is really fantastic. You feel great. You feel proud. It's part of our life. And that's why you will find all the time we are publishing and we putting in new grants. It's good. It's good. It helps. It keeps us busy. And I make people laugh sometimes. I say, we scientists, we don't need drugs to be, excited. you know, once you are doing something, you are already up there, elevated. Sometimes, of course, if the experiment doesn't work, you say, "Oh my God, what happened here?" So we, so yeah, I am very fortunate that I am funded and I can do research and help in this area.
Yeah, and that passion shows clearly in your work, because it's one thing to have the information, then there's another thing to make it available, another thing to make it easy, another thing to make it actionable. Those are all complex things that people don't realize, and you've done it. So now we're looking at pathogenic species, the bad guys.
Yes,
And you mentioned Candida a couple times. It's interesting because what you said earlier is a perfect example of the actual insight. Talk to most functional doctors and drop the word Candida, and it's straight out bad news,
yeah,
right?
yeah, yeah,
Because that's a source of so many problems. But the interpretation, the result of your research, is there's a certain level that you actually need. It's really
yes,
not Candida is bad, and overgrowth and abundance is bad, right?
yes.
And so where should somebody be on the scale?
So you can see, like, if it is maybe 1 to 2%, it's fine. When it becomes more than that, then it becomes high. And you can see the ideal range when you look at Candida. If you can see the ideal range is between 0 to 0.4%. Sometimes if you are, let's say, 1%, it's a little bit elevated, but not too much. When it becomes worrying is if it goes up. Look at this one, 21%. It's really over the top. You see what I
Yeah,
mean? So that's
yeah.
where you need to look at it, and especially this person. Not this is the genus Candida. Like if you look at all Candida together, it's 21%. If you look at Candida albicans, which is really a bad guy, is 9%, which is, again, it should be low. And the same with this respect to Candida tropicalis. So in this case, it's really, we need to bring Candida down. And the good news is, because that's one thing you will laugh at this. I started working on Candida about 50 years ago. I did
Wow.
my doctorate in England on the effect of antibiotics and steroids on Candida. And so this tells you I know a little bit about Candida. Let's put it this way. So what happens? Why do we see Candida sometimes increase? But before I go to that, I want to tell you, to mention what you already said, is that if you find Candida in the ideal range, down low, it's really very good for us, because sometimes it can break down food. Okay, it can break down food, which can be used by the beneficial bacteria as the byproducts that as their food, and then they produce a new byproduct which help Candida. So it's really complementary activity between the two. The issue becomes if somebody goes and take an antibiotic. Okay, so what you are doing with the antibiotic, you are killing the good guys, particularly lactobacillus. Okay, and lactobacillus is known to keep Candida under control. That's why first thing you want to know, are they on an antibiotic, or are they taking the anti-inflammatory steroids, for example, okay, corticosteroids? Then that is going to encourage Candida. The other factor is what their diet. If you eat too much sugar, Candida loves sugar. So you need to avoid it, okay? So to me, all of this we can put together to start bringing Candida down. And I wrote a book. It's called Total Gut Balance. It's behind me. You can see it, where we really went into the scientific background about what we call the microbiome. Remember, I said everybody talks about bacteria, which is the bacteriome, the bacterial community, but the microbiome is the fungal community. And in it, we analyzed all these issues, and we were able to come out with what we call a recommendation how to bring Candida down. And I was lucky. I worked with a colleague who is a dietitian and nutritionist, and we were able to put really recipes for people so that they can use the right diet to bring this. So we have different approaches, as you said before, the diet, the exercise, the and lifestyle included, not just exercise.
So how would somebody feel if their candida is overgrown?
There are issues with respect, some abdominal pain. Sometimes you have gastrointestinal issues. You have some inflammatory process. And really, data was published many years ago that if you have an overgrowth of candida, you have gastrointestinal issues. You have different symptoms, constipation, as well as sometimes diarrhea and other gut issues.
The unpredictable gut that people have where, hey, I ate that same food last week, but today it's acting very differently, right? Or I'm bloated, I'm not bloated, I have diarrhea, and then I have constipation.
Yes.
Yeah, that unpredictable, yeah, I get it. And we see this a lot, and here's the interesting thing, is that in our data, which is skewed towards people that are seeking help, right? People that are, for some reason, decided to get a DNA test to optimize or get better. We found that 49% of people are completely missing the GSTM1 gene, which is a primary gut detoxifier, right? Now, this again is in our data. That's not general population data. That's people that need help. So what does it mean? It's the gut is screaming to you that I am the source of disease, right?
Yes.
Right. If you feel off, it's not just diarrhea. There's a lot more going on, and especially if it's chronic and ongoing. Yeah, one day you went to a bad restaurant, no problem. But it's random, I don't get why my gut changes every day. Investigation is required. And this is where you've done a good job, because all of a sudden it's not like try this, try this, try this. It's like, no, with precision, here's exactly what's going on. Right.
Yeah. And what I love about what you said at the beginning is really you look at the DNA, you look at the microbiome, and you have a total sort of scenario how you can help people. If you find those genes that they are have issues, and then the microbiome. So when you put the total story, it's going to be really much helpful.
So why are these separated as additional species?
These because sometimes people ask us about them, ask us, do I have an increase in this guy? And also because they have some effect. Like, for example, C. diff, for example, we know that it has gut issues, okay? Like Clostridium, like for example. So we selected some of them because a lot of the time people ask us about them, and we wanted to help them with respect to that.
So you put together what you thought people need to know,
yes,
but you also added what everybody thinks they want to know. Right?
Yes, yeah, and also that's very important point you're talking about now is the fact that when you do the analysis, you have hundreds of bacteria, hundreds of fungi, in the analysis, but you cannot really share all this data with people because it's so confusing. So what we did, we tried to zoom in, to zoom on the organisms that are important, okay, and people are concerned with. So in our raw data, we have more data, but we really don't. It's not important to start to share everything because you are gonna confuse people rather than helping them.
We feel the same way. When we test, there's definitely more data we're collecting than what's provided. But we also believe that extra data is confusing. It's literally data. And then you start to say, what does this mean? What does this mean? What does this mean? For you, it doesn't mean anything. We're already telling you what you need to know. Right. And that in itself is a brilliant art of simplifying things to what's actually actionable and needed. And this is where, you hear about whole genome sequencing, and I test 10,000 genes. I test 20,000 genes.
Sure,
right.
Sure.
How many lives did you change? How many people did you help? Right. Is it actionable? And there isn't a lot that is actionable. So
I really agree with you. It's the same, exactly same like what you are talking about, which is in the microbiome analysis. Some people say, oh, we can tell you what strain you have. Like we have 10 different E. coli, 10 different. So what does that mean? It does not really help you at all when it comes to how can we interpret, how we can help you. Our job, you and I, is to try to help people have actionable insight. Otherwise, we give you all this data and then you don't, you don't understand it. Like one of the things we do in our research, we collect all this information, and we have thousands of organisms, but then we do bioinformatic analysis. Let's say we take a cohort, people who are stressed out. Okay, so we take women who stressed out between this age range and this sort of thing, and then we match them when healthy, and then we see what organisms are there. But then we do bioinformatic analysis and really identify organisms that are differentially present, which means in stress they have this handful of organisms that are really high. This is what we need to deal with. I don't want to deal with everything you have. What's important is what is the difference, and that's where really you are alluding to.
This is our community. These are people that have done DNA testing, that have had some impact because of it, and we just want everyone to know that we have been looking a long time for someone to work with on the gut microbiome, mostly because you've been asking, right? And Cindy, yes, there's going to be a replay, but we're going to get to questions. And it was challenging because everything was contextually the way you were describing big data. And I get it, because the business race, not the clinical race, is about, I want to own interpretation of the microbiome. I want to be the guy that plugs into IPM. I want to be the guy that plugs into Silicon Valley and consults everybody. That's where the big players are going. It's like, let's put some AI on the microbiome and learn things that we could never possibly learn ourselves. And that big data approach led to, here's a product that tells you got to eat more broccoli, right? And people are saying this doesn't work. And so maybe 2, 3, 5 years from now, you might find some amazing tools that AI is finding things in the big data context that points to complex diseases, etc. But the help that we can get today, you've simplified it so we can actually do that, right?
Yes.
And this is where we really love what you did. And we said there's finally someone that thinks the way we think, in the other half of personalization that we've always wanted to incorporate. And so we're doing that. So just so everybody knows, we're going to get into questions, but everyone here should know is we did decide, let's take all of this science, merge it with ours, and give you both sides of the story. And so you may have seen that the report actually doesn't say Dr. Ghannoum's name on it. It says the DNA company, right? And because we're working on taking all of his brilliant insights, all of what he knows, incorporating it into our portal, so that eventually these things meld and become one. Right? Our clinicians are being trained right now. Anyone that's in a program with us, you're going to find that the people you're working with will know what Dr. Ghannoum knows to some degree, maybe not all of it, right? And our portal will also be able to interpret, just like it is right now for DNA, what's going on in your gut. So that's the intention with all this. We wanted to announce to everybody that this partnership really gives us what we've been looking for and what you've been asking for. That's been a challenge, because it's been like 4 years of me saying, yeah, one day it's coming, because everyone's gut. And we just didn't find the right tool, but it's here. So with that, we'll tell you how to get involved, right? Anyone that's been, whether you've been genetically tested or not, it'll be available for you. Your DNA, as you know, you do once. DNA doesn't change. This is a great way for you to track. This is a great way for you to do that biannual check-in to see how much damage did I do at Christmas dinner, right? Where am I at? What work do I need to do? And this is another tool that people have been asking us for is, okay, you're telling me that I can understand my body at the personal level based on my genome, but what then do I measure ongoing to know that I'm on the right track and things are working, right? And we've introduced our DNA aging test, the epigenetics test, which does it at the DNA expression level. This will do it at the gut microbiome level. It's good to tell you a lot about nutrition, stress, all these other things that Dr. Ghannoum was talking about, so that you're always heading in the right direction. I never get off track. That's the whole purpose of this. So Dr. Ghannoum, one last time, I wanted to thank you, because, when it comes to this subject, you literally are the world leader, and we're blessed to spend this time with you, very unique opportunity. And I want to thank you.
Thank you very much for having me. I really greatly enjoyed it, and I'm excited about our collaboration, because this will bring the two forces together towards the betterment of health. And thank you for having me.
Me too.